Hey everyone, back here with part 3 of red wine processing. So let's finish off talking about malolactic fermentation. Malolactic bacteria are the most abundant microbe in wine, even more than yeast. So again, just like yeast, we have malolactic bacteria on the skin of the grape when the grapes are brought in from the vineyard. Few bacteria will grow at wine pH, and the good news for us is no pathogenic bacteria will grow. So we're making this natural product. We've got microbes that are working on our food product, but luckily, because the pH is low enough, there's nothing that's harmful to us that will grow. So that's good news. The bad news, of course, is that malolactic bacteria even struggle at low pH. So we do have plenty of malolactic bacteria, however, that will work at low pH. The lower the pH, the slower they'll go. But we are able to do malolactic fermentation. So winemakers used to consider malolactic fermentation as spoilage, but they finally realized that most wines went through it without even knowing. And I'll tell you about that in the next slide. Tartaric and malic make up over 90% of the acids in wine. So there are citric acid and some other things. But tartaric and malic are the main ones. And malic is the one that declines as we get toward harvest, because malic is metabolized when the grapes are active at night. So remember, in the last video we talked about carbonic maceration and how that's a fermentation that happens without yeast. It actually happens inside the grape itself. So similarly, when malic is metabolized in the grape at night, malic is actually consumed by the grape. So it's something that's happening internal to the grape, just like carbonic maceration. And warm nights lead to more metabolism at night, so more malic acid is metabolized when nights are warm. I said that most wines go through malic without people even knowing it. In the early days, if you were a wine cellar in Burgundy, say, that might look like something like this over on the right. The wines would be put to bed in the fall. It's very cold in Burgundy. There'd be no activity during the winter, but in the spring they would start bubbling a little bit, and the romantics said the wine is awakening and yearning to go back to the field. And what was going on that they didn't realize at the time was malolactic fermentation was starting. So there are natural malolactic bacteria in the wine, and when they got warm enough in the spring, they started being physiologically active and converting malic acid to lactic acid. So these days we usually add malolactic bacteria to the wine. We'll talk about when you want to do that, pros and cons. But you can use native malolactic bacteria. You can do an indigenous malolactic fermentation if you want. One thing is just to differentiate bacteria and yeast. I've talked about this before, but let's just quickly, both of those are single cell organisms. This is a picture of all of the three main domains of life. So bacteria, archaea, and eukarya. And yeast are part of eukarya, so they'd be in the fungi group. We are in the animal group down here, but we have a lot in common with fungi. We're both eukarya, like we've talked about before. Bacteria are way up here in a different group. Both yeast and bacteria are single-cell organisms, so they have that in common. Bacteria are a lot smaller. Yeast are quite a bit bigger, but both single-cell organisms. Okay, the main reaction of malolactic fermentation. You're taking malic acid and converting it to lactic acid. So here's malic on the left, lactic on the right. You can see that malic, or you might see here that malic has 2 acidic protons. We talked about that. Just like tartaric acid, malic has 2 acidic protons. Lactic just has 1. So lactic is a softer acid. It's not as tart. Malic acid is the acid of apples. It's much more tart. Lactic acid is the acid of milk. It's much smoother and rounder, less acidic. It only has 1 proton per molecule to give up. And you see that it has 3 carbons here, 1, 2, and 3. Again, that junction in a molecular structure like that. If no chemical symbol is given, then it means it's carbon. So there's 3 carbons in lactic acid, and there are 4, 1, 2, 3, 4 in malic acid. So when you go to malic, from malic to lactic, you kick off that 4th carbon as CO2. All right, that's why wines would bubble in the spring as they were yearning to go back to the field. It was just malolactic fermentation was starting and CO2 was bubbling out. It bubbles out much more gently than it does with yeast fermentation because you're not talking about you're talking about a lot less CO2. There are several species of malolactic bacteria. They're mentioned here. The good one that we like is Enococcus, and we'll talk about those in a sec. One more thing on this slide. The conversion from malic to lactic is done by an enzyme. Malolactic enzyme, it's called. The technical name would be malate carboxylyase. You don't have to worry about that, but just know it is converted. The conversion does happen by an enzyme. And that's true of pretty much every chemical reaction in our body, in yeast, in malolactic bacteria. All chemical conversions happen because enzymes make them happen. There was an exit card question about whether enzymes actually produce alcohol. When yeast produce alcohol, whether enzymes actually do that production. And the answer is absolutely yes, they do. So let's digress for a second, talk about that. We talked about a little bit about yeast consuming glucose, and they break down glucose in a couple of steps, and there's an intermediate molecule called pyruvate, kind of intermediate in the process. The yeast could go along, if they had oxygen, they could break down that pyruvate to CO2 plus water. And that's what we do. When we metabolize sugar, we break it down in a series of biochemical steps. And in the presence of oxygen, that's why we breathe oxygen, we would break down glucose to its to basically the lowest level, which would be CO2 and water. Yeast, on the other hand, take pyruvate. They stop at this step, oftentimes in the absence of oxygen. They could do the same metabolism as we do. They could do CO2 plus H2O like us. Yeast could do that if they had oxygen available. But in a wine fermentation, they often don't. And without oxygen, they'll break down pyruvate in a couple of steps. The first step is to break pyruvate down to acetaldehyde plus CO2. So the CO2 coming off a yeast fermentation is coming off in this step here. The next step then is to take that acetaldehyde and make that into alcohol, or to be specific, ethanol. And both steps, step 1 here and step 2, are both accomplished by enzymes. So step 1 is an enzyme called pyruvate decarboxylase. You won't be tested on that name, but look at the name, decarboxy. What does it do? It removes a CO2, a carboxy group, from pyruvate. So pyruvate is 3 carbons. Acetaldehyde is 2 carbons. And the extra carbon ends up as CO2, and it gets bubbled off out of the fermentation. Then alcohol is likewise, ethanol is likewise 2 carbons. And the conversion from acetaldehyde to alcohol is done by an enzyme called alcohol dehydrogenase. And that's how yeast convert acetaldehyde to alcohol. Interestingly, alcohol dehydrogenase, well, like a lot of enzymes, but alcohol dehydrogenase is reversible. So you can actually run the reaction the other way, and that's what our body does when we break down alcohol that we've consumed. Our body uses the same enzyme, alcohol dehydrogenase, to break down alcohol and make acetaldehyde. Then there are further steps to break down acetaldehyde into products that we can excrete easily. But when we drink too much alcohol, that's what happens. So that same enzyme, alcohol dehydrogenase, that yeast use to make alcohol in a fermentation, we use that same enzyme. It's almost identical in us. Same enzyme in us as yeast have in them. We use it. We run it reversibly. We run it in the other direction, and we make acetaldehyde out of alcohol as part of breaking down alcohol. So kind of cool, I think. But the main point here is there are enzymes that do all this conversion. All right, back to malolactic fermentation. Desirable malolactic fermentation, or MLF species. That Enococcus is the one I mentioned, is the desirable one. So here's a picture of it. These rod-like bacteria, so the individual cells here are stuck together. This picture on the right is a picture of Enococcus eni on top of a filter membrane. So if we were going to do a sterile filtration, we would run our wine through a membrane that had pore sizes that were very small, like this. And you could see these pores, these little holes, are too small, so the bacteria cannot fit through. So the bacteria would be captured on a membrane. So we'd be looking at this whole thing then is the membrane with the holes. And then these are all the little bacteria that are trapped on the membrane in this image, this electron microscope image. So it's kind of a cool picture, I think, to actually see the membrane up close and see the little pores in the membrane and how they're too small for bacteria to actually fit through. That's how we sterilize wine, is we filter wine through a membrane like this. So that's Enococcus eni. That's a good one. Here's one not so desirable, Pediococcus. This grows in unclean wine cellars, and you can get a vegetal or dirty socks smell out of it. The cells look different. Here on the right is a picture of those round cells, usually in pairs. Again, it's taken on top of a membrane. You can see the membrane holes in the background. But this is undesirable, but it will do malolactic fermentation. The next slide is lactobacillus. It'll also do malolactic fermentation, and it's undesirable. And we're seeing more and more lactobacillus infections in wineries. If you have a stuck fermentation, you're often at risk of lactobacillus starting to eat that sugar in your stuck fermentation. We would add sulfur dioxide to knock down our populations of lactobacillus or kill off some of our lactobacillus. But if you have a stuck fermentation, you do not want to add sulfites, SO2, because it's going to inhibit your yeast as well. And your yeast are already struggling. That's why the fermentation has stuck. So we have to watch out for lactobacillus. We can test for it. If we go through, if there's a decent amount of sugar around, so the lactobacillus can both consume sugar and it'll consume malic acid. So if there's a lot of sugar around, it'll eat sugar, and then it tends to produce a little more volatile acidity. So you'll get a smell of vinegar, acetic acid, or ethyl acetate smelling more like nail polish. So lactobacillus is not a good one. Moving on to flavor complexity produced by malolactic fermentation, we change our tart malic acid to soft lactic acid, as we mentioned earlier. We end up with a more creamier body mouthfeel. Again, we have a softer acid, that acid of milk, malic acid or lactic acid. So we have a little more body, a little creamier. We get a slight decrease, often in fruity flavors. Less impact on red flavor than on whites. The impact on whites is pretty apparent. Malic is a little more in the background in reds because there's more stuff going on in reds. There's more complexity, there's phenolic compounds, and there's more body in reds typically. And malic tends to not have the big flavor impact. We often select malolactic fermentation in white wines specifically for a flavor impact. And one of the main byproducts of the malolactic fermentation process is the production of diacetyl. All right, so this is produced by the malolactic bacteria. as a byproduct of malolactic fermentation. Diacetyl is that butter flavor that's used in popcorn at the movies. It's desirable in wines at different levels. Chardonnay, we typically like it. So if you've never had Chardonnay and candy corn, you know candy corn, the one we get at Halloween that has that kind of buttery flavor, try candy corn and Chardonnay. Get a buttery Chardonnay, try candy corn and a buttery Chardonnay. It's really quite good. Sounds weird? It's really quite good. And it's the buttery component of Chardonnay that complements the candy corn. So I recommend that food and wine pairing. You can select for high diacetyl producing bacteria, and you'll see if you go out to a catalog like here's something from Scott Laboratories. If you look at their catalog, you can look under fermentation and malolactic bacteria, and they'll give you descriptions of these different bacteria. So here's one that's just called MT01. It's characterized by very low VA, very low volatile acidity, and diacetyl production. And so if you don't want much diacetyl, then you'd probably want to select this particular malolactic bacteria. It is Enococcus eani, but like Saccharomyces cerevisiae, like yeast, Enococcus eani have different strains that have slightly different characteristics. So just like we can select a yeast strain to give us certain flavors during alcoholic fermentation or yeast fermentation, we can select a malolactic bacteria to give us similar flavors, or similar. We have a similar choice in selecting malolactic bacteria in order to give us certain flavors. So you can actually go out onto, I guess I don't have that. Do I have that here? No, I guess I didn't include in these slides, but there's actually a guide on the Scott Labs website for increasing levels of diacetyl, and they rank all of their malolactic bacteria on the level of production of diacetyl, so you can check that out if you're interested. But moving on, inhibitors of malolactic bacteria: low pH, SO2, scarce nutrients, and low temperature. So if you don't want your white wine to go through ML, you want to add sulfur right after yeast fermentation. Hopefully your pH is low, your acid's nice and high, and you keep the temperature low, and you can inhibit malolactic bacteria. This video here, if you want to jump out there on the web, is a little perspective on malolactic fermentation. Nothing that'll be tested, but gives you a little perspective on how one winemaker treats malolactic fermentation. We'll certainly talk about it with Pat Henderson at Kenwood next week. Here is a chart. Let's see, so this is a chart of malolactic feasibility. So if you want malolactic to go, or you don't want malolactic to go, you might want to score your wines on this basis. And you'll see that a low score, if you look down at the bottom of this chart, a low score is actually favorable for putting a wine through malolactic bacteria. Greater than 40 is extreme. You probably won't get your wine to go. And so you look at all these different characteristics. So if alcohol, if it's lower than 13, you'll have 1 point. All right. If it's up higher than 17, you'll have 10 points. So in other words, alcohol inhibits malolactic bacteria. So like alcohol inhibits a lot of microbes, and the more you have, the less likely you are to go through malolactic fermentation. Below that, if there's pH, if it's less than 3.4, 1 point. If it's less than 2.9, in other words, acid's higher, you get 10 points out of that because it's less likely it'll go through fermentation. Likewise with sulfur dioxide, if you have a low level of free sulfur versus a high level, a low level gives you just 1 point because you're more likely to make malolactic fermentation go. So this is a handy little chart, and you just total up your points here, and it gives you the scores at the bottom as to whether you'll probably get through MLF or not. So give that to you for reference. Next slide. Winemakers have trade-offs in doing malolactic fermentation. And so obviously you go from more malic if you don't do malolactic fermentation. The other end of the scale is more lactic if you do put a wine through malolactic fermentation. So what happens if you have lower malic or more malic? Well, you've got a lower pH because it's a stronger acid. You have more of a tart taste. So crisp white wines that don't go through malolactic usually are very tart, Sauvignon Blanc, etc. You get more microbial stability because your pH is lower, right? So you get more protection of your wine. Now, the flip side of that is because you haven't allowed the malolactic bacteria to eat all of, or a whole lot of, the micronutrients in your wine, you can have a little bit of microbial instability from micronutrients. So these are offsetting factors here, right? You get lower pH, so you inhibit microbes, but you also leave some food in the wine that microbes can potentially eat. So those are offsetting. At the other end of the scale, then over here, with more if you go through MLF, you'd have a higher pH. You might end up, if the wine doesn't have much acid, with a flabby taste in your wine. So you've got to consider that. You don't want that, obviously. You want a nice, tart, refreshing wine, not something that tastes like flat Coke. So you'd have to consider that. You could also add tartaric acid, of course, and still put your wine through malolactic fermentation, consume some of the malic acid, but replace it with tartaric acid. You get microbial instability if you have high pH, but again, the offsetting factor is you get more microbial stability from less micronutrients because your malolactic bacteria have consumed a lot of these micronutrients. So, some offsetting factors to think about, and you've got to think about these in light of the components that are in the particular wine. If you're worried about microbial instability in the wine, maybe you keep the pH lower and don't put it through malolactic. If you think you have a lot of micronutrients for future spoilage, maybe you do put it through in order to lower the level of micronutrients. So, just some trade-offs. Usually we drive malolactic fermentation just based on taste, flavor profile. But these are other things to consider. Do you buy bugs or do you use indigenous? You don't have to buy, but it saves a lot of headaches. It's more predictable. Here are some good brands. The bugs are sensitive, so you do have to follow directions in rehydrating them and building up the cultures, etc. You can add some directly to wine, so those are really nice. They're expensive, but they're really easy and nice to use. Some freeze-dried can also be added directly. You can definitely pitch from a wine that has malolactic fermentation going. So we did that in the fall. We took some Pinot Noir from the commercial Schoen Farm Pinot Noir production. It was undergoing malolactic, and we used that to start the malolactic fermentation in the student block Pinot Noir that we were making, the stuff we bottled last week, or earlier this week. So you can pitch if you need to. But another winemaker choice. You might get more complexity from native bugs, but it's unpredictable. When to inoculate? As soon as possible after primary fermentation, because you want to get malolactic fermentation done so you can add SO2. You want to protect the wine. You've finished yeast fermentation. Yeast, of course, when they're actively metabolizing sugar and making a lot of ethanol and CO2, they're out competing all the other microbes. When they're done, if you don't have a lot of SO2 in there, you risk other microbes growing, maybe Brettanomyces, so you want to get the malolactic fermentation done so you can add SO2 and protect the wine. You can do a co-fermentation, yeast and MLF at the same time. More and more people seem to be doing that. Some yeast strains will inhibit malolactic bacteria, so you have to be aware of that if you want to do a co-fermentation. But some people like the flavors you get from co-fermentation. Some people like the fact that you can get it all done right away and then add SO2 and protect your wine. So it's another choice. For whites and barrels, you usually inoculate directly in the barrel. So when you top barrels after primary fermentation is done, that's often when you'll add the malolactic culture. And that's where it's nice oftentimes to buy the expensive ones you can add directly to a barrel. For reds, you might finish the malolactic fermentation in tank before going to barrel. Obviously, in whites, if you're already in the barrel, you're going to do malolactic there. If it's Chardonnay, for example, some whites you might not put through malolactic, but then you have to figure out, you have to add SO2 right away, keep the temperature down, etc., so you don't go through malolactic. But reds, you're often in a tank. Before you go to barrel, you can finish MLF in the tank. Make sure it's nice and evenly done. If you let reds go through it in the barrel, some barrels may not go, and so you'd have a difference there. You can also do any acid adjustment post MLF to the entire lot, and then go to barrel. So this is, I think, not written exactly right, because if you finish in tank, you can make any acid adjustment in the tank, and then you can go to barrel afterwards. So you get a nice even distribution of that acid addition, then you go to barrel. So that is, final thing, paper chromatography is how we know if malolactic is done. And we'll talk a little bit about this when we get a chance to do this. But basically, there's a solvent that runs up the paper, and you'll spot some wines on a piece of paper, basically. So you'll have spots around here, down here at the bottom that dry. You put that paper, the bottom of the paper, you submerge in a solvent. The solvent runs up the paper, and it pulls the acids with it. And tartaric only moves a little bit. Malic moves up to about here, and lactic moves the most. And so, based on the spots or lack thereof, you can tell if there's any malic acid left in your wine. So it's a very simple way to tell if malolactic fermentation is done. It's not the most sensitive method. If you do an enzymatic method, it's much more sensitive. So you'd probably send a wine out to ETS or Vinquery to do an enzymatic analysis. But you can do paper chromatography in-house. It's cheap, it's easy, and I'll talk more about that in class. Finally, be sure it's done or take precautions. If it's not done, it's likely to happen in the bottle unless you sterile filter the wine and get rid of malolactic bacteria. So it's often not done in low acid wines in the Central Valley because they're already low in acid. So we don't want to reduce the acid further by converting malic to lactic. Again, you'd have to sterile filter those wines before they are bottled so that the MLF doesn't happen in the bottle. If you can't sterile filter at home winemaking, there are a couple of things you can add there, a couple of products that'll inhibit your malolactic bacteria. So there are, I keep saying finally, this is in fact the last slide. Red's usually done, Chardonnay almost always, light fruity reds rarely. So Sauv Blanc, Riesling, Gewurz, Pinot Gris, Muscat, not so much. Pinot Gris maybe, but the others usually not so much, especially Sauv Blanc, Riesling, Gewurz. These low pH tart wines won't go through ML typically. Sparkling can go either way, winemaker choice. Okay, so we're going to leave it there. There are a few slides left in that slide deck having to do with Pinot Noir and barrels and some of the sensory characteristics. I'm not going to cover those here. We'll touch on those slightly in class at some point, but don't worry about those for now. Thank you very much for listening. Have a great weekend, and we'll see you in class next week. Bye.